This is a production of Cornell University.
Thank you, Nina, and good afternoon, everybody. Thank you for joining me today for the seminar. So to give you a little overview of what we're going to be talking about today, I'm going to talk a little bit about, just very briefly, research in botanic gardens in general. I'm getting the Master's in Public Garden Leadership, so I want to kind of give a perspective on that. And then I'm going to jump into some research I helped out with at the Morton Arboretum in Chicago last summer. And I'm going to finish up by talking about some of the research I've been involved with while here at Cornell. So in botanic gardens, there's been a history of research that's dated back about 500 years now. With the earliest iterations of what we'd actually consider a botanic garden, there was a research focus at these institutions. The first kind of botanic gardens that we really considered seeing in the world were what was known as the physics gardens. And these were gardens that were associated with universities and usually primarily were focused on teaching students about medicinal plants and for doctors who were studying. So the first gardens to be established were in Italy at the University of Pisa. And even from back in this date, we see that research has been an important part of what they do. Stepping into the more modern day, some of the research that we see that has kind of traditionally happened at botanic gardens has really been work mostly around systematics. So looking at phylogenetic relationships of plants and classification, particularly with plant exploration and the work that was going on of bringing plants from other parts of the world and classifying them, as well as floristics. Floristics being looking at plants from the perspective of biogeography and the distribution of the different species and what affects speciation in this way. And this work has, of course, been supported by herbarium work, the drawing of physical plants to do this kind of taxonomic description, but then also increasingly with living collections. So bringing seed back or living plants and growing them on in collections and using them for this type of study. Kind of some more modern focuses that we're starting to see more gardens put an emphasis on is definitely conservation biology and the disciplines that go on within that. Chicago Botanic Garden and Missouri Botanic Garden being two examples of gardens that have a strong focus on plant conservation and the associated fields. And of course, horticultural research has also been an important topic that botanic gardens have played a very active role in. Like I mentioned, particularly with plant exploration, going to new parts of the world, bringing plants back, and then with that, subsequent plant introduction. And when we grow these plants on in our living collections, we also gives us an opportunity for plant evaluation, such as disease screening or pest screening. And this is in many ways tied into plant breeding programs, and really gardens have started to become more of just a nice place to go visit and see plants from around the world. But we're starting more and more to consider them as what we would consider a germplasm repository, where these wild collected plants are representations of their wild populations, which can then be used for study, conservation, or plant breeding, as I mentioned. So that was a very brief overview of research in botanic gardens. And with that, I'm going to transition in to talk a little bit about the work I did at the Morton Arboretum. For people who aren't familiar with the Public Garden Leadership Program, between your first and the second year, all the fellows in the program go to somewhere in the United States and participate in an internship. I found myself at the Morton Arboretum for a number of reasons. They've got an excellent plant collection that I was very interested in interacting with. But the Morton Arboretum, to give a general overview, is 1,700 acres, so quite a large garden. And it's more than just the Arboretum. They focus on more than plants. They have natural areas, as well as prairies, a number of formal gardens too, such as a children's garden, which is a great way to diversify your gardens to get different types of audiences to come and visit. And they also have some other specialized gardens, like a hedge garden and fragrance garden. They have a $27 million annual budget, employ over 350 employees, including part-time and seasonals. And this garden was established in 1922 by Joy Morton, and people will typically know the Morton name as the Salt Empire and people that, in that industry. And at the Morton Arboretum, they've got many thousands living plants, but they have 4,100 different specific taxa, so different species or subspecies growing on the ground. And the Morton Arboretum was an interesting place to go because of their living collections and because it is such a widely visited garden. but they also have a very well-developed research program at the Morton Arboretum. And they focus on a broad number of disciplines, from plant systematics, some of our more traditional research that we'd find in gardens, to forest ecology. Actually, Bob Fahey, who's the son of Tim Fahey, who some folks might be familiar with in natural resources, works there. They also focus on plant conservation, arboriculture, root biology. And the lab I was most interested in working with was the urban soils lab. So Dr. Bryant Scherenbrock heads up the Mass Soil Science Laboratory. And what makes their lab a little bit interesting is that they have a number of different areas of research interest, but they really focus on urban soils and urban ecosystems as their central focus, such as in urban ecology and biochemistry, and assessment and remediation of urban soils. They also do some focus on forest management as well as prairie studies. And these last two areas, the prairie ecology and forest management, is actually a lot of the research that gets done physically on the grounds at the Morton Arboretum. So that's something that makes them a little different as well, is that they use their landscape and view their landscape as a living laboratory. And to give a kind of overview of what Bryant's lab looks like, because it is a little different than what we tend to see in the academic setting, is that Bryant has been there for 5 years. He's published 12 publications and done 53 presentations at this point. But one of the major differences is that there's only 2 employees in the lab, his lab manager and himself. And he works with graduate students, but not specifically as chairs for committees, more kind of tangentially, like the way he's worked with me. And he does sit on some committees. But where he kind of makes up the difference in productivity that graduate students will bring to a lab is in his use of volunteers. So in 5 years, he's ended up having over 150 volunteers in his lab, as well as 41 interns.
It appears that some of his volunteers are quite young.
Yeah, and he'll enlist anyone that he can, including his children, to do the lab work. And this is something that is, volunteers are something that Botanic Gardens heavily use to help fulfill their work and their missions. And this is just one example, because it's an interesting one, of how his lab can still be very productive, but in a slightly different context than what we may be familiar with. So jumping into urban soils, which is going to be what we talk about for the remainder of this talk, I'm going to read this definition because I think it really sums up what urban soils are. Urban soils are non-agricultural, man-made surface layer, more than 50 centimeters thick, that have been produced by a mixing, filling, or contamination of land in urban or suburban areas. So really what we're talking about here is we're talking about soils that aren't agricultural, aren't found in a natural ecosystem, and typically in one way or another are adversely or are impacted by human use or human presence. And why is this an important area of study? It's an important area of study, particularly in the United States, because in the next 40 years, we're projected to have 5% more urban areas in the United States alone. And humans have unique impacts that alter the soil's biogeochemical processes and really make them a distinct and unique system. So in surveys of urban soils that have been done, we tend to see a suite of characteristics that we will find, such as high bulk densities, decreases in organic matter, poor structure, high pH, low water holding capacity, decrease in aggregate stability, inadequate soil depth for root growth, and a decrease in microbial biomass. So when I was at the Morton Arboretum, I specifically helped out with the Chicago Urban Forestry Study. And the Chicago Urban Forestry Study was a joint study between the Morton Arboretum and the U.S. Department of Agriculture. And there was 2 main PIs on it. It was Bryant Scherenbrock in the Urban Soil Science Lab, as well as Bob Fahey in Forest Ecology. I'm primarily going to be talking about this first area. The second area, Robert Fahey was mostly using dendrochronology techniques to look at how the urban forest was responding to differences in different climatic events, so particularly drought. He wanted to see how the forest was responding to that. And the justification for the study is that there had been no comprehensive study of carbon storage that had been conducted in an urban system based on different land use types, and then looking at the soils both in a shallow, 20 centimeters, as well as deep, up to 1 meter in depth, and in some places deeper than that even. And this is important because in the urban environment, Soils themselves make up the single largest terrestrial sink of carbon, with up to 64%, followed by vegetation, which are two factors that we considered in this study. So looking at about 84% of all the carbon that cycles in the urban system. In a study that was conducted in 2000 by Jogaby and Jackson, they looked at 3 different natural land ecosystems, land types, and looked at how carbon was distributed within the top centimeter of soil based on these three land types. And what they found from this initial study was that in shrublands, you had a larger percentage of carbon distributed in the deeper part of your soils on one end of the spectrum, while in forest lands, you ended up with carbon primarily in your top 20 centimeters of soil. And this was mostly explained in the shrubland systems as by how deep the roots were going and turning over in the soil. And with the forest, like in deciduous forests with leaves or coarse woody debris accumulating, you find a higher carbon in the top 20 centimeters. So Brian kind of looked at this idea and thought about where he could move forward with it. And the 2 types of carbon that he wanted to look at in these systems were organic carbon, as most people will be familiar with, or carbon that's derived from a living source, either plant or animal, and living and dead microorganisms. But he also wanted to look at inorganic carbon, which in soils typically refers to carbon found in a carbonate form, which is derived from mineral parent material. And in urban systems in particular, inorganic carbon is more of a question of protection than it is about sequestration. With all the disturbances that go on in urban ecosystems, by exposing these inorganic carbon deposits, you can actually create a source for carbon into the atmosphere versus a sink, as we may think about with our organic carbon. So there's a number of factors in particular that affect carbon cycling within these soils. And what I want to point out here is that all the ones that I put stars next to, so from here on down, are different factors that can be affected by land management or by just how we use our land or our presence on it. And so carbon cycling within the shallow part of our soil is primarily derived from organic carbon. We see high rates of turnover here, and these are sensitive to human management and typically found in your organic or your A horizons. Where deeper, beyond 20 centimeters, it's a slower turnover rate. You find more of your inorganic carbon sources, and you can also find organic sources, and this is where you start to find your A horizons, your E horizons, and your B horizons as well. So to understand the framework of how the Morton Arboretum wanted to construct the study looking at carbon based on land use, they went back to a study that was conducted in 2010, also supported by the Morton Arboretum. In 2010, the Morton Arboretum conducted a tree census of the Chicagoland region. And what the tree census was is they selected the 7 counties around Chicago as well as the city itself, and they generated 2,076 random plots. They went out to each of these plots and they looked at, they characterized the plots based on 2 sets of criteria. They looked at the plot information, so what was the land being used for, as well as percent tree cover, shrub cover, and the shrub species. And then also when they found trees, they would look at the trees. Specifically, they would characterize the species, the stem diameter, tree height, crown height, and its general health, how much missing foliage or dieback had occurred, crown light exposure, and as well as distance to infrastructure or other buildings. And they took all this information and they used it in the iTree model, which for people who aren't familiar with this, is a model created by the USDA and used by urban foresters or city planners to quantify the structure of urban forests within a municipality town. And what this model does is, one, it gives us a breakdown of all the different species and how much canopy cover perhaps you're finding within your city, but it also puts terms, it puts it in terms of ecosystem services derived in a monetary value. There's going to be a way to justify having an urban forest and maintaining it and diversifying it. So they used this model and created all this data. And so Bryant took this information and they decided to, for this carbon study, to take a subset of these different land use types. So these are all the different land use types that we looked at in the greater Chicagoland region that we wanted to assess for carbon cycling in those systems. So we took a number of different types of samples we took deep samples using this, it's hard to see in this picture, a large soil sampler that takes samples down to 1 meter in depth. We took shallow samples down to 20 centimeters, as well as our litter layer, whatever was primarily on the top of the soil. And we brought these back to the lab, and we'd look at each of the deep soil profiles based on horizons and characterize them based on color, pH, texture, bulk density, soil moisture. We would also take our litter extracted, and we would set them up and extract any of the invertebrates as maybe an additional part of the study that they'll be looking at in the future. So we looked at carbon in 3 different ways. We looked at our inorganic and organic carbon with elemental analysis and gas chromatography. And we looked at our organic carbon particularly in 2 ways. One is particulate organic matter, and the other one is using active carbon. I'm going to talk about these 2 tests. So particulate organic matter is a way of quantifying and looking at organic matter based on size. So what you do is you take your organic matter and divide it from your sand silts and your clays, and you fraction them into different size classes. Each of these there's 4 different size classes. 3 are represented here, the other one being smallest organic matter that's associated with your silts and your clays, so your organo-mineral complexed organic matter. And the theory goes is that depending on the different size classes, each size class has a different residency time in the soil, with anywhere from our litter soil organic matter for being less than a year to our organo-mineral complexed organic matter being anywhere from 400 to 1,000 years. Bryant had done a previous study in the past where he looked at time since disturbance of sites, so how long it had been since a site had been in an urban ecosystem, had been kind of created or established, and looked at particulate organic matter and found that this coarse particulate organic matter, particularly on the 1 to 2 years, was a very good indicator for microbial biomass carbon, as well as nitrogen mineralization. So this information, if used, could be helped to use and figure out nitrogen calculations for street trees, as the theory went. So this is one way of looking at organic matter that the Sharon Brock lab is very, very fond of. And the other technique that we used was the active carbon test. So for folks who are familiar with the soil health test here at Cornell, this is the preferred method that they use. It's a potassium permanganate oxidation process. And this has really started to become in favor for looking at our labile organic matter, our organic matter that turns over quickly in the soil, because it's really easy to conduct. It's on a colorimetric scale. And they've known that it's sensitive to changes in management techniques. So we see that it correlates well with basal respiration, aggregation, and microbial biomass. And so all this information that the Sharon Brock lab is working on is going to be cycled into the iTree model itself and used to build a carbon model for soils based on land use type, just like the iTree model had been used to create ecosystem services for above ground biomass. So that about sums up the work that I did at the Morton Arboretum or was involved with. And I'm going to transition now to talk about the research I've been doing while I've been at Cornell. So for folks that saw me spoke earlier this year, what I've been working on while I've been here, I've been working with Nina Bassick and Don Rakow and Harold Van Ness on the scoop and dump technique of soil remediation. And this technique is pretty much just what it sounds. We come in, we take compost, we put it on impacted soils or compacted soils at a rate of 6 to 8 inches, and then we come in with a backhoe, physically lift it up, and drop it. And what this creates is a heterogeneous soil mixture that landscape plants can be directly planted into. And then after that, after the planting, mulch is added every year, as you would find in most landscaping contexts. And this technique was developed based on 2 kind of studies. The first one being a study done in 1991 by Kai Rolfe, who came in without adding organic matter, just using a backhoe to physically fracture soils. And what he found when he did this, looking at soil quality, he found that soil bulk density decreased, that pore volume increased, and soil resistance, so using a pentrometer in that soil, also decreased. Secondly, a study finished up in 2007 by Nina Basic and Angela Rivenshield looked at using different types of organic amendments and seeing how those affected soil bulk densities and soil porosities. And that's how we came up with using this 33% organic matter as an ideal amendment. So the scoop and dump technique has had a legacy of practice on the Cornell campus we're going on to year 14 now at this point. Every year in Nina Basic's Creating the Urban Eating class, this is a year-long course in site establishment of plants and woody plant ID. The class will design, create, and install a new garden. And we'd been doing this technique for more than 12 years when I started, and it has been used to restore sites such as some people will recognize this. This is outside of Mann Library when it was under construction. And severely impacted soils occurred during this process. And here's a picture taken even a couple of years ago of what the landscape looks like now. And another one. So we had seen that anecdotally, we knew it was working. We had some research to back it up, but we wanted to take it another step further and really see what the changes in soil quality had occurred based on using this technique. So in the first part of our study, we selected 6 sites representing 12 years to be studied in our scoop and dump soils. And we selected 4 sites that were in close proximity to our study sites that had gone through similar disturbance regimes and could be used as a control or as a contrast. And we used the soil health test to look at physical, biological, and chemical aspects of the soil in our initial study. And the way we sampled is we would dig into our scoop and dump soil, and we would sample that, and then we'd go below our scoop and dump layer into our subsoil, and we'd also sample that. And then for our controls, we would take samples at equal depths to our scoop and dump as well as our subsoils. So looking at soil resistance, so coming in with a calibrated pentrometer and doing it all shortly after rainfall, so very close to field capacity for soil moisture, we saw that in our scoop and dump soils that we started to see root-limiting PSI, or root-limiting resistance, I should say, in our scoop and dump soil right around 25 centimeters in depth. Compared to our unamended soils, we found them right around 9 centimeters in depth. So about a 3-fold increase in available root depth that we were finding in our remediated sites. As I mentioned, we look at this study in 2 ways. We looked at it in a compare and contrast with our unremediated to our remediated sites. We also looked at it over time since we've been doing this practice for years. And over time, we saw that bulk density decreased over time, with year 0 being over here. We also noticed that we found that our potentially mineralizable nitrogen had increased over time, as well as our active carbon. And this was exciting to see and certainly makes sense with the potentially mineralizable nitrogen and active carbon being a food source for soil microorganisms and organisms that are involved in nitrogen mineralization with the potentially mineralizable nitrogen increases we see. And the bulk density increase was also exciting to see, particularly with our increase in aggregate stability. We were seeing that we were creating lower bulk density soils, but they were also being maintained by this improvement in structure, improvement in pore space. So to summarize the other findings we found from this initial study, we found also increased aggregate stability. We found increased available water holding capacity, increased total organic matter, increased potentially mineralizable nitrogen, bulk densities, as well as active carbon. And this is kind of a common scene of what we would find in many of our sites. This site had only been installed 1 year when we looked at it, but this is a profile difference between our scoop and dump soils compared to the subsoil. And you can see, we found this all across the site that once we get down to that subsoil, which had higher bulk densities typically, we would see the roots just start to move sideways instead of continuing to move down. So we took all this information and we wanted to think about how plants were grown in this type of environment and do a study to follow up what we had learned in our first section. So with that, we came up with a soil container study that we wanted to conduct to look at how plants themselves would respond to these different soil mediums. So we hypothesized that plants grown in the scoop and dump soil will have increased biomass, and this increased biomass will be correlated with our reduced bulk density, our improved bulk density. And then we also hypothesized that because of our increased water holding capacity, the plants grown in our scoop and dump soil would have a better capability of dealing with drought, particularly in stomatal conductance as well as photosynthesis compared to our unamended soils. So we took a subset of the sites we used before. We selected 3 sites, and we selected 3 controls that went along with them. And we went out into the landscape, and we took large PVC cores, and in this case we had to use a backhoe to drive these into the ground and pull them up. And this is the best available technique we have at getting as close to undisturbed soil samples as possible. It's perhaps a slight overestimate, but it's the best we work with. We didn't have to use a backhoe in our scoop and dump sites because the bulk density was decreased, so it was a little easier to sample. And so we selected a woody plant to grow in them. We selected Ficus benjamina. And we ended up using Ficus benjamina, one, because it's known in tropical regions of the world for being a street tree, as we can see in this picture here. And we also wanted to use it to get around the winter dormancy issue if we had selected a North American temperate species, which would have been ideal, but we were working over the winter. So we selected Ficus benjamina evergreen and had tissue cultured plants donated to us from Sunshine Horticulture in Florida. All a pretty consistent size when we got them. So we collected these soils outside. We brought them into the greenhouse for a short period and planted the ficus directly into the different types of soils. And shortly after that, we moved them into growth chambers and allowed them to grow for 3.5 months before we decided to conduct our drought study. In the end, we ended up using 15 soils that had been remediated and 15 soils that had been remediated with the scoop and dump and 15 that had not been remediated for our study. And we subjected them to a period of 26 days of drought. And before we started the study, what we did is we got the plants up to field capacity. So we brought them up to a high point of water holding and then let them drain and then wrap them in bags so that really the only water that would be lost is through transpiration and not evaporation. So our measurements that we wanted to look at in the study were root and shoot biomass, total leaf area, and leaf water potential at pre-dawn. And the soils characteristics that we wanted to look at were gravimetric water content, volumetric water content, and texture, water holding capacity, total organic matter, and bulk density. And in terms of gas exchange, we looked at photosynthesis as well as stomatal conductance. So what we ended up finding from our study was that our leaf area in our scoop and dump soils over here was greatly increased, almost twofold compared to our unamended soils. So much more leaf area. We found that our root dry weight was significantly increased in our scoop and dump soils compared to our unamended soils, as well as our shoot dry weight was significantly higher in our scoop and dump soils compared to our unamended soils. We also looked at bulk density, since this has been a major question of our study. And we found that in our scoop and dump soils, they were significantly lower than our unamended. And for folks that aren't familiar with bulk density is a measurement of weight over volume, so grams per centimeter cubed. And a study done by Dado and Warrington had established a relationship between different textural classes and root-limiting bulk densities. And what they found is that for your sands, you can get up to bulk densities around 1.75 grams per centimeters cubed before you end up finding reduced bulk densities. And for silts and clays, it's around 1.4. So considering, again, that our scoop and dump soils were well below this 1.4 threshold, it was very good to see. And then our unamended soils are definitely getting into the range of root-limiting bulk densities. And we don't have the textures back from our study yet, but we'll be able to look at those compared to these once those final tests are completed. So we did our linear regression looking at our dry weight shoots with our bulk densities, and we see a clear relationship here. And if we overlay this with our different types of treatments, our scoop and dump soils on the left here, and our unamended, we see a clear relationship between the bulk density and this improved scoop and dump creating, allowing for more shoots to be grown. And this line here represents root limiting bulk density, really strikingly kind of cutting them right down the middle there. So we saw this relationship as well with our roots. Here's it with the overlay again. And we also saw this relationship with our leaf area. And again with this overlay. And so when we destructively harvested these, we obviously noticed certainly a much larger volume of roots in our scoop and dump comparatively to our unamended soils. But one pattern that was very consistent, here are pictures from our unamended sites, was you can see from how I'm holding up these roots right here, is that in our unamended soils, right here is kind of where we planted, where the plug was that we planted into the soils themselves. And a very consistent pattern where the roots growing up across the top and then escaping down the sides of the containers, right down here, in an area that had perhaps a little extra space in it or did not have the same type of bulk density as we found in the soil. So this was a very, very consistent pattern, and one that's interesting to think about as we, since we did this in containers in the landscape, roots probably wouldn't have this, or they certainly wouldn't have this container or reduced area where they would be able to go down. So we actually think that our roots are a bit of an overestimate compared to our scoop and dump. It was also very interesting to look at the root exploration in our scoop and dump soils. We really saw that our roots were really exploring and taking advantage of this increased structure with our aggregate stability and able to find those pore spaces and really work their way through. And it was interesting pulling them apart because it would take a lot more work to break up the aggregates and actually free the roots from this system, compared to in our unamended soils, where pretty much you'd start to break it apart and the soils would just all fall away. The plant was not holding very much onto them because of this decrease in structure and root exploration throughout the soils. So looking at our leaf water potentials, this is days of drought on the bottom here. We see that as in both the unamended and the scoop and dump soils, that really there wasn't much different in how these plants were responding to drought in terms of leaf water potentials. And we certainly saw a strong spike after day 16 of our potentials starting to get really negative at this point. And this was a fairly consistent pattern that we saw with our photosynthetic rate and our gas exchange measurements as well, where both the plants, there was no significant differences in over the course of the dry down experiment, showing a difference in photosynthetic rate, as well as stomatal conductance was also had no significant differences between the two. And if we started to think about this and look at this, we also looked at, like I said, water loss of the soils themselves over time. And the red up here is scoop and dump soils, and the blue is unamended, and this is a cumulative water loss over time. And with this, what we see is that the scoop and dump soils did in fact hold and lose more water compared to our unamended, which was corroborated with our increase in available water holding capacity, where we lost 0.098 kilograms of water over the course of 26 days, while about a little more than half of that in our unamended soils, just 0.65 kilograms of water loss. And looking at our water loss between days, we also saw that as there were significant differences between days 1 and day 16 in terms of amount of water loss between days, with our scoop and dump soils losing more water compared to our unamended, and then that difference no longer occurs after day 21 and day 26. And so looking at all this, it starts to make more sense that even though our scoop and dump soils held more water, and we did see an increase in both root growth as well as total leaf area, that there was an increase in still monoconductance, an increase in transpiration, which really seem to explain driving this pattern between our different soil amendments. And with that, I'd like to thank my committee and everyone else who's been a part of my research progress here, as well as the Morton Arboretum, and I'm happy to answer any questions.
I just wanted to make sure I was understanding. It seemed to me like you had two types of comparisons in your study of the scoop and dump soil. So one was the unamended versus the scoop and dump, but then also the scoop and dumps against each other over time.
Right.
So okay, it was so quick that I didn't quite catch it. So you saw increasing potentially mineralizable nitrogen and active carbon and decreasing bulk density the longer
Over time.
Oh, that's so interesting. So, so what do you make of that?
Well, I think the bulk density certainly makes sense with the previous work that Kyle had done in terms of loosening the soil and maintaining that loosened soil, creating more pore space. And it also, thinking about with the increase in aggregate stability, once the soil had been loosened and aggregates were starting to form, that increase in structure certainly makes sense in terms of holding more pore space between the different aggregates and creating a less bulk environment. In terms of the active carbon, I think that would certainly make sense with the huge amount of organic matter that we input initially, as well as the continued adding of organic matter creates an environment where more I guess, preferential for microbes to live. So
it kind
over time, as we continue to add more organic matter, we're seeing improvements in that. That makes sense.
So it kind of seems like that initial remediation sets in motion kind of a virtuous cycle. Like once you have better aggregation, it can sequester more carbon and
Certainly,
so
yeah.
forth. So,
Yeah.
Oh, because I would expect the immediate difference between the two treatments, but that increase over time is very interesting.
Well, that's something that you do tend to see also in, like I mentioned, the study that Brian and Sharon had done before, we looked at time since disturbance, that after, I want to say around 25 years, you start to see soil starting to cycle back in kind of a natural cycle. So even without doing too much to your landscape, you end up with improved soils without having to, without doing something drastic.
I have a question about that same part of your study. So did you have the same species in all of these landscapes? So clearly your plant material would affect your soil characteristics as well, particularly if that landscape has been established for some period of time. So were you always sampling near the same species or in an area with no plants, or how did that work out?
We ran transects through our plots and then went at certain intervals through each one. So we weren't as conscious of looking at individual species. This first study was more of just a survey to look at what, really what the changes in soil quality had been. And so looking at in a more fine-tooth comb at, 13, 14 years ago, in retrospect, it would have been great to have said every year we're going to plant one single species in all of that. But we didn't do that, so we didn't quite have the option. And that was something that we did think about trying to look at in terms of looking at some of the plant responses, since they all received very similar management over the years, but we couldn't find any selections that were the same, because part of the reason we create these landscapes is to create diverse learning opportunities for the students in the class. So, in maybe a more traditional context where we have a lot more of the same species that we find in our kind of normal landscapes outside of how diversity is here, that was an option. That wasn't an option for this study.
If the figs in the dump soil had higher transpiration rates and larger leaves, why wasn't the water potential lower? And why wasn't photosynthesis lower? They're drawing more water out of the tank.
Yeah, I mean, well, I think it makes sense if we think about the total leaf area in terms of still monoconductance at least. We had greater leaf area in our scoop and dump soils, almost twice as much. So that was driving transpiration.
Well, that should have blown off more water and made the soil drier.
Right.
because of
But okay, so our soils had more. Even though they had more leaf area, we had higher water holding capacities in our student dump soils. So yeah, I think that makes sense.
So I had a question on a totally different topic that you also touched on. So you talked about the importance of volunteers in the Botanical Gardens research. And I'm really interested to know what you think about if and how the principal investigators in contexts like that can create a valuable kind of educational and or professional development experience for those volunteers.
For those volunteers.
'Cause I mean, I could see how it could be, very much a kind of grunt work thing where you don't get much out of it, but I can also see how you could shape it to be something really transformative.
Yeah, and that was interesting about Brian's lab in particular, because of all the labs at the Morton Arboretum, he took advantage of volunteers like anybody else. And the type of people that volunteered were really across the board from, like he got his kids in there. Maybe they didn't want to volunteer, but more
No choice.
From people who were in college looking to gain experience to he had retired chemists in his lab who would actually come and just wanted to do something with their time and take their skill and put it to use. And so for him, it's become a very effective tool. And also he's been able to tap into a lot of resources, and because of having these people with such depth of knowledge in different types of fields, they bring all sorts of resources to the lab and allow them to do things like that. So I don't know exactly, but it's at Botanic Gardens there's this kind of tradition and history and practice of using volunteers.
Sure.
So it's a great benefit to see, and I've tried to think about how to apply it into other contexts, because Botanic Gardens are these mission-based organizations that people are often used to going and visiting and having a kind of experience that, and it's a little different than what we see in the academics. So it'd be great to see that kind of thing there.
Well, thank you very much.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.